Well, thank you for giving me the opportunity to speak at this wonderful event. I'd like to start by saying that my talk's going to be a little bit less overwhelming than some of the other talks, because this is really a very early technology. It's taking the first baby steps, but I hope I'll be able to enthuse you for the potential of where we can go with this. So I'd like to start with, I think this molecule doesn't need any introduction to this audience. This just to point out a few points that will become more important later in the talk. So nucleic acids have a tripartite structure. I don't know, is the laser, there is it. Tripartite structure, nuclear base, sugar scaffold, and phosphodiester linkage. And these arranged in an aperiodic polymer have the ability to form these canonical antiparallel helices with the amazing ability to store genetic information and thereby enabling evolution. And evolutionary strategies clearly have precedence to be this successful in drug discovery. All the antibody drugs are made either by an evolutionary process within the animal, within the human body, or by phage display, which is another evolutionary technique, and obviously MedImmune is here who've been really based on that technology. But what I'd like to talk about is really evolutionary methods as applied to this type of molecule, because really nucleic acids have the potential to be much more than just molecular memory. And in particular, single-stranded nucleic acids have the potential to fold up into three-dimensional structures. Okay, doesn't want to play. Okay, so this is, it clearly doesn't want to play this. So this is the protein structure of thrombin, and here behind it you see the thrombin aptamer in blue. And all I meant to show in this movie really is that aptamers, which are single-stranded nucleic acids folded up, can fold up into these three-dimensional shapes and really bind to proteins and any other type of molecular target in very much the same way as antibodies can, with the same sort of specificity and affinity. As you can see, hopefully here with this sort of intricate, you can see the aptamer intercalating into the intricate molecular surface on the protein surface. But aptamers really have underperformed in the clinic. And to show you one of the reasons, this is the one aptamer which has so far been licensed, Macugen, in the US for treatment of macular degeneration. So this is directed against vascular endothelial growth factor. But because aptamers are discovered initially as RNA and DNA strands, they're rapidly degraded inside the body. So they need to be modified by medicinal chemistry. And this just shows you the examples that had to be done to Macugen. Pretty much all the pyrimidines had to be modified with a 2' fluoro position. And all the purines, except those two A's here, had to be modified with 2' methyl. But these two A's couldn't be modified because that would actually affect or destroy the biological activity of the molecule, thereby leaving an Achilles heel for degradation. And really, this type of iterative medicinal chemistry process post-discovery greatly increases the cost and the time required to develop these drugs. And also, as you see, leaves potential Achilles heels and may affect affinity and specificity. So one of the approaches we took is to try to expand the chemistry of these compounds. So this is a type of nucleic acid we've been working with. HNA stands for anhydrohexatol nucleic acids, because the 5-membered ribofuranose ring of DNA is replaced with a 6-membered hexatol ring. And what this does is it makes the polymer that results completely impervious to degradation by DNases, RNases, or in fact we can throw the whole New England Biolabs catalog at these molecules. They won't be degraded, and they essentially in serum they last forever. They'll last for weeks in serum. They also are impervious to depurination, which would happen to DNA exposed to low pH. You can see exposure to pH 1 and then pH 9, as you would passage through the digestive system, DNA goes very rapidly, while HNA is much more stable. But there's a problem because HNA is not a substrate for natural polymerases and therefore not compatible with the type of discovery technology that we need to make aptamers. And our approach has been to basically develop new strategies to engineer polymerases so we can apply evolutionary technologies to HNA and, as you later see, to a wider range of these modified nucleic acids, genetic polymers. So we use three main technologies to expand polymerase function. We use structural data, and I don't need to make the case again after Lawrence's excellent talk, how important it is to understand the structure, to understand function. This, in fact, just shows a heat map of where in the polymerase we need to mutate to acquire the ability to use HNA as a substrate. We use evolutionary methods based on water in oil emulsion technology, and we also try to mine phylogenetic data to better understand polymerase function. And putting all these together, we've been able to build polymerases that can now read a DNA strand and synthesize the HNA complement, and also take the HNA as a template and reverse transcribe it back into DNA. So clearly, DNA and RNA can now interchange genetic information, just as you can do between DNA and RNA through the use of polymerases and reverse transcriptases. And in fact, we can now, akin to a retroviral replication, we can now set up a replication cycle based on a DNA intermediate, and that enables evolution. And the technology can be expanded to you can see we can expand it to other 6-membered ring analogs. This is cyclohexanyl rings, tetrose sugars, cross-linked 5-membered rings, and arabino sugars. And we'll now be able to apply this to evolutionary cycle. So this is how we can drive aptamer discovery. It's a very simple method. We can start with simply a random pool of sequences. You can order the DNA templates from any commercial supplier. The polymerase will convert them into a random pool of HNA sequences. We degrade away the DNA using DNase and fold these up into 3-dimensional shapes, and then bind them to a solid phase target. The non-binding sequences get washed away. The binders remain bound to the target. We can elute the binders, reverse transcribe them back to DNA, and start the cycle again. And this iterative process will ultimately enrich from a very large 10 to the 14 molecule sequence pool binders to specific targets. This takes just a few weeks, and I should show you two very simple examples. The first one, nucleic acid target, HIV TAR RNA motif, important for the viral life cycles, and there are a number of RNA aptamers that have been made. This shows the structure of an RNA aptamer binding the HIV TAR. All the aptamers discovered, or most of them, bind the RNA motif through this kissing loop interaction where you get a hairpin aptamer binding this sort of final loop of the TAR motif. This just shows what we got. So this just shows a quick little bit of data. Specificity analysis, these are different TAR RNAs. The magenta parts are scrambled, and as you can see, the RNA aptamer will bind all the RNAs that retain in green the final loop motif, while the HNA aptamer that we've made actually requires the whole motif for recognition, and the same selection also gave us another one that is specific for this bulge. Affinities are typically in the order of what you would get with phage display in a sort of first round of phage selection, mid-nanomolar binding. Maybe more challenging protein targets. So this is Hennig lysozyme, another model target, just to show you again specific binding to Hennig lysozyme, cross-reaction with human lysozyme, which is closely related, and similar affinities in the mid to higher nanomolar range. And maybe what's most important, these binders can function in complex environments. So this is a plasmacytoma cell line, which genetically modified to express Hennig lysozyme on its surface. As you can see, these can pick out the Hennig lysozyme expressed on the surface in the context of all the other membrane proteins with good specificity in FACS. And just to finally show you how tough these things are, so I don't know if you're familiar with biocore binding curves. So this just shows the association and dissociation rate of the aptamers, and the signal is the sort of binding amplitude. And what you should be able to see is two curves, one a dotted line and one drawn out. And these compares the aptamer binding before and after treatment for 3 hours at pH 1 at 40 degrees, followed by pH 9. And you see not only do the association and dissociation kinetics not change, also the binding amplitude is completely unchanged. So these binders are completely impervious to this really rather harsh chemical treatment. So to just summarize, so we've been able to take heredity and evolution beyond DNA and expand the chemistry of nucleic acid polymers that can be synthesized, replicated, and evolved. You can see we've expanded this now to 8, and I think we're up to 10 now. We're able to make aptamers, and we're currently working on making these to more clinically relevant target. We'll be able to make, we're able to make enzymes as well, and also these nucleic acids have this convenient ability through to the predictability of Watson-Crick binding rules to be assembled into three-dimensional nano objects, devices, and structures, and I think these might have a number of applications in biotechnology. And just to finish up, I mean, as I said, these are tripartite molecules, and really all parts are available for chemical modification. So in the future, we hope to explore this, what we'd like to call the XNA space, chemical space. So we've concentrated initially on changing the sugar chemistry, replacing it with other ring congeners, and this, as you can see, has quite profound effects on physicochemical properties and stability. We've also done work on, you can attach chemical group side chains, if you want, to the bases, which can confer things like increased hydrophobicity or fluorescence and so on. And finally, we're working on changing the phosphodiester linkage to take out the charge, because I think one of the key objectives for future work will be to be able to make nucleic acids more suitable for reaching intracellular targets. I think this is where they should have an opportunity to compete with antibodies for applications in the clinic. And I think I'd just like to end by thanking the great people who did this work. So what I described today really is just the work of 3 people, Vitor Pinheiro, Alex Taylor, and a great student, Chris Cousins. And I thank you for your attention.